So it is my delight to introduce Dr. Mark Mandel from the University of Wisconsin in Madison. He's in the Department of Medical Microbiology and Immunology. So Mark, he ended up earning his bachelor's at Cornell, and this was in biology, and that's really where he first started studying microbiology, and he was actually working on Wolbachia in children. So then, as a PhD student, that's where he became really a bona fide bacterial geneticist in the lab of Tom Salhavi at Princeton, and he was working on RPOS regulation in E. coli at that time. So I really got to know Mark during his postdoc years because I was a postdoc and we shared the same office at UW-Madison in Ned Ruby's lab. And so we overlapped probably for at least a year and a half, 2 years, something like that. And what I highly recommend, if you haven't done so, Mark had a Nature paper in, I think it's 2009, and that remains one of my favorite papers, just overall. And he highlighted this signaling protein that was taken in by horizontal gene transfer into Vibrio fischeri, and it gave Vibrio fischeri the capacity to colonize squid. So it's just a fascinating read. Really took off right when comparative genomics was taking off. So yeah, I highly recommend checking out that paper. So then what's remarkable is Mark ended up getting a faculty position at Northwestern during the Great Recession when all of these jobs were just disappearing. So, that just highlights the caliber of a researcher that Mark is. He ended up getting tenure as an associate professor and then moved back to Wisconsin, same department, and bizarrely in the exact same lab that we shared when I was a postdoc there as well. So that's just a very surreal experience seeing the same lab that you're in as a postdoc. So let's see. So yeah, Mark, it's been great to reconnect with you. And yeah, we're looking forward to having you here at Microbiome.
Our appreciation for you coming. We have a small plaque.
Thank you so much. Tim, thanks so much for that introduction. Really appreciate it. I think I'll pause for a second to say, one of the things that's really nice about science is how you have these connections over years, over decades. So I think the person in this room who I met earliest was Seth, actually. So it was 25 years ago. I've done the math on this 5 times, it's correct. Graduate student Seth came down from Rochester to Ithaca and gave us some input on this Wolbachia project that I was the only one in the lab working on Wolbachia in the context of these field cricket hybrid zone. It was just super useful to have that input and to have these connections over such a long time is just incredibly fulfilling and add something to what we do beyond the science, the training. That's just I don't know. It's really.
Keep the stories about Tim and I to yourself.
Oh, there's one in here, but it's pretty mine. Okay. All right. So today we're going to talk about bobtail squid. I know a lot of people know this from Tim's amazing work and that of people in his lab, but I'm still going to give a little bit of background for people who are less familiar with that. Okay. So these are our bobtail squid. They're about the size of golf balls. They're out buried in the sand during the day. They come out to forage for food at night. And they would cast a pretty nasty shadow or silhouette in that moonlight, except that they use counter illumination. So they have luminescent Vibrio fischeri bacteria. They colonize a dedicated organ, the light organ, that's only on the underside of the squid. And light from the bacteria camouflages that shadow from the squid. And it's modified to the amount of light that's coming from the moon. So on a full moonlight, a lot of bacterial light comes out. And on a cloudy night or a new moon, very little light comes out. Okay. So it provides some advantages. If you picture, for example, a dolphin with a black dorsal side and a white ventral side, there's not a lot it can do to modify that. Whereas here you have a lot of, you can have communication between the host and the symbiont to have a sort of dynamic system. And actually, back to the comment that I said earlier, if people have questions, feel free to jump in with them. I've got a small section at the end of the talk that I can include or not include based on how we're doing on timing. All right, so valuable features of this system for understanding microbiome development. I'm not going to read through all these, but basically we can rear the host and the symbiont separately. As Tim mentioned, my background is in bacterial genetics, so we do a lot of bacterial genetics. There we can make mutant libraries, we can make precise mutants, we can change amino acids within the bacterium, we can fluorescently label the bacterium, and then introduce those carefully constructed strains back into the host and ask what they do. Recently, the host genome was sequenced, and there's still a lot of annotation going on there. And we're hoping soon to be able to manipulate the host genetics. So Josh Rosenthal's group at MBL at Woods Hole has recently done some CRISPR approaches on the host and made some, for example, what they're calling albino squid, where they've removed all the pigmentation from it as a proof of principle to be able to do this. So the fundamental question that we're seeking to address here, though, is how is it when this animal's born into an ocean with 1 million bacteria in every milliliter of seawater, it gets colonized in this one organ with a monoculture, right? How does it pick out Vibrio fischeri, which are not that plentiful in the ocean, and fill this one organ with that species to the exclusion of everything else that's in the ocean? And as you'll see as we go on here, it's not just Vibrio fischeri, it's actually only certain strains of Vibrio fischeri. So a little bit on the practical nature of this. So we collect these animals from nearshore waters of Hawaii twice a year. You can see these are pretty shallow waters. We go out where it's a shallow reef for quite a ways. We collect them, and I have a couple of videos to show what it looks like when we're collecting them. So sometimes the squid's on the bottom of the ocean, and we scoop it up with a net right from the sand. So here's the squid right in the middle. It's burying in the sand here. And sometimes you'll see just the eyes poking out here. We play a fun game called, is it a squid or is it a rock? Sometimes we're wrong. And then sometimes we'll have the squid just swimming right in the water column. And see that right in the middle there? And then you can just sort of net it right in the middle. So about half of them we find are in the bottom and half are in the middle. And if you had told me this is what I'd be doing when I became a bacterial geneticist, I would have been very surprised. Right, so we bring them back to the lab. We have them in tanks that probably look very similar to what Tim's look like. We raise them here, we mate them in the lab, they lay eggs. This is what the eggs really look like on the top right. And then this one, they're all matted with sand that kind of glues them to the undersurface. We flip them over, you can see the eggs themselves, it's about 400 eggs here. They incubate for just under a month. They hatch in the lab. When they hatch, they lack their symbionts. And so that's why they're so useful to be able to then introduce our lab bacteria to. So that's sort of the normal process. But if you're a lab at Penn State, then sometimes squid spend a night at the Days Inn in Sandusky, Ohio on the way, because we deal with a lot of sort of issues with shipping and logistics. There was this one time about 10 years ago where squid that were supposed to end up in Pennsylvania ended up in our lab in Chicago. And then it was hard to get them here. So they took about a 10-hour drive across Ohio. We dropped them off in the Days Inn. Andrew picked them up at the Days Inn. And you can ask Tim for more details about that. Okay, back to the science.
Question.
Yeah.
Besides the light symbiont, are there other microbes that may already be in the new hatchlings that come from mom or the environment?
Yeah, so the question is, are there other microbes in the new hatchling? So there's definitely ones in the digestive tract that sort of seem to pass through. There's been, no one's done a careful study, but we don't know if there is actually a true sort of stable microbiome there. Spencer Nyholm's group has done work on this jelly coat in the egg that seems to produce antifungal compounds, protects the egg throughout that. Whether any of those are actually transmitted to the squid is not clear.
Okay.
Yeah, I think it's an open question.
Cool, lots to learn.
Yeah. So on the left here, we sort of have one adult here and then a bunch of kind of teenagers. This is what in a bowl in Hawaii. When they hatch, they're Drosophila sized. So here they're in a 50 mil conical tube. It's a couple hundred in the tube. If we zoom in on one of the hatchlings on the right side is under a dissecting scope. And just to give some sense of the anatomy here, so here are the eyes. This ski cap structure here is the mantle. Water then flows through the mantle cavity over the gills so the squid can breathe, and then it gets pumped out here. What's highlighted here is the ink sac, and then just below that, closer to us, is the light organ that you can't really see here, but you'll see on the next slide. And as water gets pumped through here, this is how environmental bacteria come into contact with the light organ. So if we zoom in on this with this micrograph, now you're seeing mucus that the squid is producing, and that's how it begins to trap the bacteria. And this is the first level of selection. As you get this incredible environmental diversity gets pared down to the specificity within the host. Okay, so bacteria, and especially gram-negative bacteria, get preferentially entrapped within this mucus. On the next slide, I'm not going to be showing you the mucus, but I'm going to show you the bacteria, which are expressing GFP. And what you can see on both sides of the organ here, we see an aggregate of GFP expressing bacteria that are about to swim into one of the pores on either side of the light organ. Okay? So this is just 3 hours after we squirted them into the bowl. Okay? And so 3 hours after, every squid is going to be colonized with Vibrio fischeri. It's super efficient. What you can see is that the bacteria have to aggregate, and they make this biofilm-like aggregate on their way in. And that's going to be pretty important as we go on. If we now fast forward to 48 hours, what we see is that the bacteria are really tightly packed in one of the squid crypts. They're right up against the epithelial cells of the host. And this is basically the stable association. So they're like this most of the time. At dawn, the host will expel 95% of these cells. The remaining 5% grow up during the day. They make light for the host at night using form sensing. And then that process ensues for the lifetime of the host. But the acquisition of the bacteria and where sort of the specificity occurs, where they acquire new bacteria, that really only happens during the first week or so of the new squid hatchling's life. So we want to understand this process of how does the host select these bacteria? What are the bacterial behaviors that are important for this process? Really understanding that initiation symbiosis is really important. All right, so as a cartoon version of what I told you so far, we squirt the bacteria in a bowl. They start to encounter the squid, and they form these biofilm aggregates at 3 hours. And then they enter these densely packed host crypts, and they form these more stable associations. And I'm showing you a 48-hour time point, but it sort of happens gradually in these daily cycles over the first week or so, and then that persists that way for the lifetime as well. Okay, so now to get in the first story here, what are the genes that are required for this reproducible colonization process? And this is something that was tackled by the first two students in the lab, John Brooks and Denise Levitt. So the first part I'm going to go through quickly because it's all published. The other two stories are unpublished. So we use transposon sequencing to identify genes that are critical for this colonization process. And so briefly, we took a 50,000 mutant bacterial library. We introduced it to the squid, and we identified genes that were either underrepresented or mutants that were underrepresented or overrepresented afterward. And so there's the orange one as an example here of one that was underrepresented. So we inferred from this that the gene that was interrupted in this mutant was required for the colonization process. Or we have this sort of green one, which was overrepresented after. And so we hypothesized that this represented an inhibitor of the colonization process. If we remove the inhibitor, we get more of that mutant represented afterward. And there is a bit of a bottleneck into the squid. But one nice thing about the system is we can do this in pretty large numbers. So we did this in 6 batches of 250 squid each. And so what these genes are, this is the protein chaperone DNAJ in the orange. Well, we found that many bacteria DNAJ is essential. It's not essential in Vibrio fischeri. There's actually very little even growth defect for it, but DNAJ is required for that symbiotic biofilm formation. And it does this through the canonical pathway of taking proteins to the ATP-dependent DNAK. It does that. We have some information that does that. We're still interested in learning more, but it does impact a lot of pathways of Vibrio fischeri. So we haven't gone much into the mechanism of how that works. The green cartoon here is a hybrid histidine kinase called BINK. And what we found here was that if we inactivate this kinase, we get more biofilm formation and better colonization. And I'll show you some figures of that on the next slides. So here what I'm showing is we get those aggregates of bacteria on the way into the squid. If we make a clean deletion in BINK, what you can see is that we get much larger aggregates. And if we complement back a wild type copy of the gene, we restore the regular aggregate size. And we can capture a lot of these squid phenotypes with certain wrinkled colony assays in culture, which allows us to do things like do bacterial screens, do them rapidly in culture, and take them back into the squid. And so a version of this assay in culture looks like this, where we lose the inhibitor and we get expression of that biofilm in culture. We can complement it like this. And then if we mutate, for example, the histidine that's required for phosphoryl group transfer, we no longer lose that. So from this, we infer that it's using the typical two-component system phosphoryl transfer. And we have other evidence to support that as well. There's a really neat trick we can do with this. So if we lose BINK, we get more biofilm. And if we overexpress BINK, we can actually shut down the biofilm here. And so there's another version of that wrinkled colony assay up here. And it's actually so powerful that we overexpress that inhibitor here. This assay, we're looking at squid colonization. We can actually inhibit the ability to colonize squid. And so on the next slide, I'm going to take you through the signal transduction pathway for the biofilm formation in the squid. So everything on the left here was worked out by Karen Weissig's lab at Loyola University Chicago. And I'm just going to go through it quickly, but then I'll tell you which actually gene names you need to know. So there's a hybrid histidine kinase at the top here that starts off the signaling. It then passes the phosphoryl groups eventually down to a transcription factor. This turns on this 18 gene locus that makes the exopolysaccharide that turns on the biofilm that's required for colonization. And I've shown you now two different assays. One is this wrinkled colony assay, and the other is the aggregation of the host. And just to be clear, these are the same outputs. Okay, these are the outputs of the same pathway. So when we overexpress this top of this pathway, it makes more of this locus, and that allows it to make the biofilm in culture, and it makes more of the aggregation here. If we mutate just about any of these genes here, it blocks this phenotype, and it blocks the squid. So there's this incredible close connection between the phenotypes we see in culture and those we see in the squid. But to simplify this now a little bit, okay, this is what is important to remember here. RSCS is the positive regulator. CYPF is this sort of hub protein here, and all that turns on biofilm and host colonization. Okay? And we can forget the detail for the rest of the talk. So I just told you we identified this negative regulator BINK, and this is where it sits also in the inner membrane, but on the cytoplasmic side, okay? And it inhibits the pathway this way. So now we have this positive regulator RSCS, this negative regulator BINK, and presumably somewhere in this pathway, it's interpreting host factors to make these decisions. If we look at the big picture, Vibrio fischeri is swimming as single cells in the ocean, and then it hits the squid, and all of a sudden, within a couple hours, perhaps faster, it changes from the solitary lifestyle to this group behavior, right? And to do that, this is the system that does it. We don't think CYPF is doing it because actually you only need the cytoplasmic portion of CYPF, this last domain. That's the only part that's required. So our initial hypothesis was either the squid is turning on positive regulation of biofilm or it's de-repressing negative regulation. And we wanted to figure out how can we get at that question. And so when Denise took this over for her grad work, she had a clever way to do it. She said, okay, we've got this sort of seesaw here, this positive and negative relation. Maybe they're canceling each other out. And so normally RSCS is required for colonization. If you take this away, you don't get colonization. She said, is that still the case if we also remove the negative regulation? So if we take off the brake, do you still need the gas pedal, or can you coast in the squid? And that's what she set out to test. So this is the squid colonization assay that I breezed through before, but I'm going to explain in detail here. We're looking at the number of bacteria in the light organ of the squid. We allow the bacteria to colonize for 3 hours. We then move the squid to new water, and then we assay the number of bacteria at 2 days. What you can see is that the wild type strain colonizes really well. We get 10 to the 5th bacteria in there. If we lose the negative regulator, it's no problem. It's still colonized just fine. We lose that positive regulator RSCS, though, they really do not colonize. This is an aposymbiotic control, so there's no bacteria here. So what happens in the double mutant? What you can see is they colonize just fine, right? So it seems like the idea of that positive regulator is it's overcoming this persistent negative regulation that's there from BNK. Okay. I do want to add a caveat. Yeah.
Is that an otherwise healthy squid though, or have the bacteria sort of taken over in a pathogenic way?
Yeah, we don't have great markers for what's going on in the squid.
Yeah.
I mean, so I'll say two things about that.
Okay.
First of all, we know there's still a function for RCS there. So if we compete this strain, okay, against wild type, for example, if we compete this strain against the Delta BNK strain, we still see a role for RCS. Okay? So it's clear that it still has a role, even a strain lacking the negative regulator. The second thing is this strain alone that lacks the negative regulator, there are some, I don't want to say fitness issues, but there's issues we can identify. And that's even though we identified this and that the BNK mutant can outcompete wild type getting in, we think the bacteria kind of get stuck in there. And we think there's probably a transmission defect for that strain. And even though these BNK mutants have this incredible competitive advantage to get in, we never identify BNK mutants among natural isolates. And so we think there probably is a problem over the full life cycle of the symbiosis. Yeah.
Yeah, right. In the last slide, you mentioned there is a middle factor in the pathway.
Yeah.
I don't remember the name, but
yes,
did you test the involvement of that? Is it interacting with the negative or the positive regulator in any way possible to control the colonization?
So if we remove CYPF, you lose the ability to colonize. But if you have just that C terminal domain of it, you get all these, most of these same interactions.
Okay.
That changes a bit when we stop just looking at one strain and widen the lens, and I'll have a section on that later on.
Okay.
Yeah.
I'll look for that.
Because when we remove it, they can't colonize, it does constrain some of the questions we can ask with it. But maybe we can come back to that. Yeah.
Thank you.
Okay, so to get at some of these questions a little more, we want to know, like, are they just in, or, I mean, a bit to Seth's question about, like, is it a normal symbiotic interaction or is something off? Everything we can measure, though, on the bacterial side, at least, seems fine. So one question we asked then, are they making normal sized aggregates on the way in? And they're fine. So this imaging of those aggregates, we still see that sort of the extra large BNK size aggregates compared to the smaller wild type aggregates. That's quantified here. The other thing that Denise started to look at was she looked at biofilm gene expression. So now these are not just labeled cells. This is a fluorescent reporter that's connected to the biofilm locus. Red is background just to identify where the cells are, and the green is the expression of this. And this is actually Tim's reporter that's been super useful. So thank you very much for that. What you can see is even on agar, where we get like really great sensitivity, you can see that whether or not you have RCS there doesn't matter. This was sort of baseline for this, and now we want to take it into the squid. And even in the squid, we can see is that whether or not you have RCS here doesn't matter. And we see a difference on the baseline here at 48 hours, whether or not you have BNK. But in that background, where you already don't have the negative regulator, having RCS or not doesn't matter. Okay, so I'm going to tell you now why that matters. So the question I set this up as is, where are the host signals coming in? Before we identified BNK, the model had been that the host signals through this positive regulator and turns on the system. When that's the only player we had, if we took this away, everything, we couldn't get colonization, so we couldn't ask these questions. But now, in the background where we remove BNK, we can say plus or minus this factor, the bacteria still aggregate the same, turn on gene expression the same, and colonize the same. So it seems like they are not interpreting a host signal through the left side of this pathway as to whether they should be planktonic in seawater or aggregate in the host, right? So it seems like our current model for how this is working is that the positive aspect of this is constitutive. And if we want to look for where they're getting a signal from the host to change their behavior in response to symbiosis, it's going to be coming in through the right side of this path, right? And so that's our current model, is that they're basically, BNK is keeping us off in seawater, and they hit the host, and then the host says, you're in the right place, it's time to stick together, and that turns back off. And so naturally that led us to ask, what is the host signal? Was there a question in the back? No, okay. And so Natalia Rosario Melendez took this over as a master's project in the lab. She made incredible progress given that this was interrupted by COVID. And Denise, who stayed on the lab as a scientist, has picked this up since. And so again, this is the pathway as we're thinking about it now, asking what is the signal that's feeding into BNK? And so a little more on the structure of BNK. It has two transmembrane domains with a periplasmic cache domain, which is often known to bind small molecules. And then it has the two component signaling domains in the cytoplasm. And so the approach that Natalia took was to purify the cache domain and to look for ligands that could bind. So we did a sort of fairly small scale screen for this. If you're familiar with biolog phenotypic microarray plates, you can order about 8 of them. So it's about 750 or so compounds. And we did this process called differential scanning fluorimetry, where we take the purified protein, bind it with Cipra orange, and then put it through a melting curve on a real-time PCR machine. And in each of those wells, we've added one of those biolog compounds. And then look at where the curve changes, basically where binding of the compound is changing the unfolding profile of the protein. I'll zoom in on one of these in a second, but basically Natalia looked at these for every compound in those 8 plates we looked at and looked for a shift in the protein. And so what you can see is for ethanolamine, she saw about a 10-degree shift in that melting curve. And in black is the protein alone. And for other amine compounds, we didn't see any shift. So this seemed to be specific for ethanolamine. In the set of compounds we looked at, there were actually 2 copies of ethanolamine, and they both yielded that shift. So one downside to using this as a screening strategy is that you don't really have the concentration of the compound. So we validated this with a known concentration. We could see this. Then she did a microscale thermophoresis to calculate a KD for this and saw something of 144 nanomolar that seemed to be biologically relevant. And around the time we were doing this, Wei Long Eng's lab at Tufts was also studying the cholera quorum sensor receptor CQSR, which also has a cache domain. What they had found was that their cache domain also binds ethanolamine. And I have to say they've been really helpful and supportive as we've gone on this. What they had found was that theirs was about 50% identical to BNK. And there were 2 aspartates that are required for binding ethanolamine in their domain. And so we went on now and mutated those aspartates in BNK. And when we do that, we lose activity. So I'll just show one example here. So BNK is a negative regulator. So you lose BNK, you get more biofilm. You bring the wild type copy back and you restore the no biofilm state. But that's not the case if we mutate one of the aspartates. You get the same phenotypes here if we're looking at biofilm gene expression, right? More biofilm correlates here with more biofilm gene expression. And we're still getting expression of the protein here. So this Western blot against BNK, you still see the same levels of BNK even when we've mutated that residue. Okay, so what happens in the squid now? When we mutate, here's either of those aspartate residues, we get the same thing where we get more biofilm. So we basically failed the complement in those cases, and we can see some of those here. We get larger biofilm, larger aggregates in the squid. And so our interpretation of this is that it's binding to a compound at that cache domain. Whether it's ethanolamine or something like ethanolamine is something we're still working out. So we've tried, for example, if we add ethanolamine itself. So if the model is that the squid provides ethanolamine and that says, hey, you're here, it's time to aggregate, the expectation would be if you provide that compound, that then you would get more aggregation, and we don't really see that. And so we're considering two different models now. One is that it's a related compound, and that's the true signal from the host. The other is that its bacterial provided ethanolamine is constantly interacting with that receptor, and that the squid interferes with that signaling. And we're actually favoring that hypothesis right now. So we're trying to identify what is it from the squid that might play a role in interfering with that sort of constitutive signaling. If anyone has ideas how we could get at that, would be really exciting. Okay. So getting back at the question that Rim raised earlier, how does this pathway of RSCS, CYP as the hub protein, MINK, how has that evolved to mediate host specificity? So you've heard Tim mention work during my postdoc about the protein you're talking about was RSCS. So RSCS is really present in a number of North Pacific Ocean Vibrio fischeri strains. And basically, if you have it there, you can colonize. And if you don't, you can't. There's a lot of fish isolates of Vibrio fischeri that don't have RSCS. They can't colonize, but if we put RSCS into them, they can. But what we started noticing as we were sequencing and analyzing Vibrio fischeri from around the world is that correlation didn't always hold. And so I've been talking this whole time about a Hawaiian strain called ES114, and it has this pathway of this positive regulator, this negative regulator that turns on biofilm and allows host colonization. But there's one strain in particular that we were looking at, this Mediterranean squid isolate called SR5, and it did not have this positive regulator, RSCS, but it still colonized its Mediterranean squid host. And if we took it to the Hawaiian squid, it could still colonize the Hawaiian squid just fine. And so we want to know, does it use this pathway at all? I mean, maybe it has some completely other way to get inside the squid. And so this is something that I think I skipped over the most important part here. So this is something that Ella, who is a postdoc in Chicago, worked on, and Katie, who's a grad student in Madison, tackled for her thesis. Okay. And so the first question was, does it even use the same biofilm pathway at all? And the answer is yes. And so this is the Hawaiian isolate on the left here, ES114. This is if we delete the regulator RSCS or the target CYP biofilm genes, doesn't colonize. SR5, which doesn't have the regulator, but if we delete the biofilm target genes, we get some variability in this assay, but you see it has at least a 2 log deficit in the ability to colonize. And so we want to ask, how does it do that? And so to approach this, okay, to be fair, we did 5 other things before we did this, but I'm going to zoom ahead to this. Okay, so for this, we asked, is the information for this encoded within the locus itself? Okay, so all the stuff I told you didn't need to know earlier, some of it becomes important now, but you didn't need to know until now. So CYP-F, this turns on the CYP biofilm genes. And it's this 18 gene cluster. And CYP-F is one of the genes that's encoded within that locus. Okay. And so there's 3 regulators within that locus, CYP, E, F, and G. They're all 2 component regulatory genes. And so we asked, how similar are they? And CYP-E is 90% identical to amino acid level. CYP-F is 96%, and CYP-G is almost 100%. And so, you know, our best candidate here was CYP-E because it was the least similar. But because they're all next to each other, we just swapped them all. And for everything I can tell you about this work that Katie did, these are all precise allelic exchanges in the native chromosomal locus. And so this is, I just want to say it's a lot of work. But we felt we had to do this because when we tried putting this on a plasmid, or we even tried putting it in a neutral site in the chromosome, we would get weird phenotypes. And what I mean by that is things that would not complement if we did it, or that would not get the same phenotype in the same, let me be precise. When we do these wrinkle colony assays, the colonies that would be smooth, if we would put that gene back instead of at its native site, and we would put it at the neutral TN7 site of the chromosome, we would get a little bit of wrinkling. And what that told us was that the amount of gene expression was not quite the same if we put it here or here. And so we felt to test this hypothesis, we really had to be doing this all as an apples to apples comparison. And so what Katie did was she swapped these two loci, okay, and then asked, what are the phenotypes if we do this swap? And so the first question was, what happens now if we just take them two from ES114 and from SR5? And so in ES114, the Hawaiian strain that has that upstream regulator, that RCS, it doesn't matter which set of regulators you have. You have the Hawaiian CPFG, the Mediterranean FG, it's totally fine. But it's not the same case for the Mediterranean strain. Okay, so for the Mediterranean strain, you need the Mediterranean CPFG. If you take the Hawaiian CPFG, now all of a sudden you drop your colonization ability by 2 logs. Okay? So it seems to matter what's downstream of that. Okay? And now we do the reciprocal experiment. Now we're taking the Hawaiian strain, and we've deleted RSCS in this strain. So now it kind of looks like the Mediterranean strain in that sense. And now if you have your Hawaiian regulators, you no longer can colonize. But if you take your Mediterranean regulators, now you can colonize without that upstream RSCS, okay? So this is the same phenotype here. If you have a strain with without the upstream regulators, you need the Mediterranean CPFG. And now you have one without RSCS, and you need the Mediterranean CPFG, okay? All right, so now let's figure out, is it just one gene that's doing it, or is it the whole set? And it's just one gene. So really here, once you lose CYPF, the middle gene, you drop out, okay? And so it's necessary to have the Mediterranean CYPF, and it's also sufficient here. Into the Hawaiian strain that doesn't have RSCS, if we bring back only the Mediterranean CYPF, that's sufficient to allow colonization without the positive regulator. Okay, so back to this model here. What I've told you so far is that both strains use the same biofilm targets. Okay? Both strains require CYPF for colonization, okay? But it's not the same CYPF between the two. Okay? And so if you have a Hawaiian strain, okay, you can have any CYPF here and it allows colonization. But if you do not have this upstream regulator, either because in evolution you never had it, or because this is what you have now, because we've deleted it artificially, then you need a special CYPF, right? There's something different about the Mediterranean CYPF that allows them to colonize. And so we wanted to ask what that was. Yeah.
Could you recapitulate colonization just with F?
Just with F. So in what background?
In the previous assays that you did. So yeah, here, if rather than expressing the whole cassette, if you just express F.
Yeah, so that's what this experiment is actually. It's a little hard to tease through. So in this assay, we have the Hawaiian strain where we've deleted the upstream regulator, and we've just—
Oh, it's just one. Okay, okay, I thought it—
Yeah.
Okay.
So we've swapped just the CYPF gene and just the open reading frame.
Got it.
Into its native locus in the chromosome. Yeah. Oh, we got that great animation again. So that's good. Okay, so this is like, you know, a 900-ish amino acid protein. And so the next thing that Katie did was to sort of break it apart. And I'm just going to focus in on where the key difference is here. So what we're showing here is both those in-culture biofilm assays and also squid colonization. And the difference here is, so in purple is the Mediterranean part of the protein, and in blue is the Hawaiian part of the protein. And what you can see is that if you have the 2-component receiver domain from the Hawaiian protein, you don't make the biofilm and you don't colonize squid. But if you have it from the Mediterranean part of the protein, then you do stimulate biofilm production in colonize. So that one domain of the protein is required here to do this. But this is a little convoluted because you have other parts of the protein that are different. So we wanted to just isolate that one part of the protein and really ask if it was sufficient. And so that's what she's done here. So here, what we're showing is that here's the Hawaiian CypF. It really does not induce biofilm gene expression or the wrinkle colony in the way that the Mediterranean allele does. But if she swaps in just that one domain, that's sufficient now to induce that independent of that upstream. And we now have colonization data to support this. Okay. So what this is telling us is that there is sort of this is the model I showed you before. There seems to be this really conserved host biofilm module that extends beyond these Hawaiian isolates, right? That's, determines whether or not you can colonize squid. And it predates, I'll show you a tree in a second, but it predates whether or not they acquired this upstream regulator. Okay? And what we think we're seeing is that these convergent pathways, and so in the Mediterranean, the way that they have figured out how to colonize squid is that they have evolved an allele of CypF that has, let's say, an overactive rec domain. And what we think is happening is that it's putting CypF in a kinase tilted state. And so that's leading to more phosphorylation of the downstream CypG and allowing that to make biofilm. In contrast, the way that this has evolved in Hawaii has been by horizontal gene transfer, acquisition of RSCS, and then that's feeding in phosphoryl groups in that manner. And so I'm not going to go through everything on this tree, but in general, this is a fischeri tree. These are the two strains I showed you about, the Mediterranean strain SR5 on top, the Hawaiian strain ES114. The big difference I want to show you here is that this is acquisition of RSCS here. Most of these strains in the gray can colonize squid, right? So getting that regulator was like a big hammer, right? And basically then you can colonize squid, and the only way you lose the ability to colonize squid is if you have a big change, right? So you have you then get a horizontally acquired something that lost you the ability to colonize after that. In contrast, the Mediterranean strain is kind of the exception. Most of the ones in this basal part of the tree are not squid colonizers. They're fish colonizers. They're seawater isolates, okay? And so we're now looking to trace how has this occurred, and are there other ways? We think there probably are other ways that have evolved. And so what I've shown you in this last part is sort of the ancestral group, and especially non-Vibrio fischeri. For the combinatorial logic of how this works is that you might have part of the pathway, but maybe you have non-functional biofilm genes. And then when Vibrio fischeri came on the scene, almost all the Vibrio fischeri, perhaps all the Vibrio fischeri we had, has this functional biofilm target locus. Okay? And then in those Hawaiian isolates and Japan squid isolates as well, they acquired RCS, and that turns on those biofilm targets in a developmentally appropriate way in the squid. But then if we look at that basal group, they may have the targets, but they're still not getting turned on unless they've evolved a secondary way to do this, or a convergent way to do this. And the way that this one Mediterranean strain has done that is by having this hyperactive CypF. What time do we end? Okay, I'll go like 5 more minutes then. Okay. So something we've learned from these studies is that these small genetic changes can really lead to profound niche differences, right? If your point of view is that you're a bacterium in the ocean, and what animal hosts are available for you, where can you colonize? I think it's really interesting that, you know, a change in a regulator. One point I didn't make is this domain difference is 3 amino acids, right? This is a really small change that has this profound effect on where you can live. And so we've now seen 2 differences. So acquisition of a gene in Vibrio fischeri, or these 3 residue change in CypF has this big difference in where you can live. And there's other examples too from the symbiosis world. So Heidi Goodrich-Blair's lab showed that these 3 gene acquisition in Xenarabis nematophila can change host specificity and which worm species are available for you for symbiosis. There's a couple of really cool examples from Yersinia pestis in terms of flea midgut colonization and other aspects of their lifestyle. And what I think is interesting about these is that they often relate to biofilm formation as well, right? So this balance between, are you living a solitary or a group lifestyle, has a really big difference on which hosts are when you turn those pathways on. So you're a microbe in the world. Where can you live, survive? These have big impacts. Can you engage in these group behaviors? Can you do them faster than your competitor in a way that it makes a big impact? How do you interpret and respond to these cues from your environment? And how does this evolve? In the last 2 minutes, I'll just say something really quick about how do you interpret those cues from your environment. And this is a project that we've taken on the last few years that Denise has worked on. And we've done this in collaboration with Laura Sanchez at UC Santa Cruz and with Terry Moore at University of Illinois, Chicago. And our squid model is a really nice size for trying to get it in situ chemistry. And we want to ask, what are the compounds that are at that symbiosis interface? And so we've started this in colonies. And these are the same biofilm colonies I've been showing you. So it's a little hard to see on these matrices, but this is basically non-wrinkled, a little wrinkle, and like a lot of wrinkling. And Laura Sanchez's lab uses mass spec imaging to identify in an unbiased way, what are the small molecules that are in these? And so what her group did was they found a whole bunch of compounds that were enriched when the bacteria made more biofilm. And here we've got the mass to charge ratio for example, the sodiated addict is at 257. And then they went on, this is like the slide that they used to, when I asked my dumb chemistry questions, this is the one that they used to say that they can use tandem mass spec to break these masses apart and rebuild structural information from them. And then what her and Terry did was they identified a candidate diketopiperazine from this, which is like 2 cyclized amino acids. And this is a histidine and a proline that are together. And then they used chiral chromatography to figure out the orientation of these. And this is D-histidine and L-proline that are together. And remember, this is when bacteria make a lot of biofilm, they make this. So you can sort of think, if you're in a genetics world, you can think of this in the regulon of biofilm. And it's not just the genes that are activated, but the chemicals that are activated as well. And these family of compounds, they have varied roles in microbes and in hosts. But what they do is like really nebulous. It's not clear what the receptors are, what effect they're having. And so we started investigating what does this do in Vibrio fischeri. And we found that it had an effect on bacterial luminescence. And so when we add it to our squid isolate, it has a little bit of an effect. This is a fairly dim strain that we work on. So we took a brighter strain and we could see a bigger effect. And if we look at one time point, for example, you can see that it has over 100-fold effect at the 100 to 250 micromolar range. So we're using this as a way to build this technology and to now say, we've done this in colonies, what can we do in the animal? And so I'm just going to jump to the end here. I sort of had a little bit of how we did this, but this is the animal now where we've flattened it. We've done it in the same matrix and now looked in a spatial way and said, unbiased way, what are the compounds we can see? And then segregated those regions within the animal to say what regions give similar chemical profiles. And this is sort of our third attempt. We're still building up here. But what we can see is that there's this green region here, which corresponds to the light organ and ink sac region. And we get about 150 compounds that are unique to that light organ. And one of the things we're really excited about was that, a little hard to see, but if we overexpress biofilm, we can see that diketopiperazine in there. But even in the wild type, if we're not playing any genetic tricks, we can still detect the compound in that region. Okay? And so we're excited to use this in the future to try to identify compounds directly from the symbiotic interface, perhaps that we would not detect in the culture, and hope to get emergent biology that we can only get from studying that host microbe. So I think I've acknowledged folks along the way as I've gone. really grateful for the collaboration that's been valuable for agencies, and I'd love to take additional questions.
A few minutes for questions, and as usual, we appreciate the first question coming from the trainee. Or up then to everyone.
In the nature, do you see that squids are always colonized with just one strain, or is there a mixture of strains?
Yeah, so the question is, in nature, squid colonized with one strain or a mixture? It's a mixture. So people are fairly surprised by that result. So I think probably the best data on this is in about 15 years ago, Michael Wollenberg did a study where he took a lot of adults and then looked at strain diversity within them and found that there was about like 6 to 10 different strains within each. And there were often, I'd say, 2 profiles of strains within them. And for example, there'd often be a set of brighter strains and a set of dimmer strains within them. We don't really understand what's going on there. To what forces are shaping it. Yeah.
You take a Mediterranean strain and a Hawaiian strain, do they co-colonize, or what happens? Does it depend also on what water you take, like maybe more salty water or less salty water?
Yeah, it's a great question. If we compete a Hawaiian strain and a Mediterranean strain, a Hawaiian strain will outcompete that, but we are always doing these in Hawaiian squid. So we'd like to do that in the native host as well. I don't know. And actually, another answer to your first question, in the Mediterranean squid, there's often a separate species of Vibrio as well. So Vibrio logii is a sister species to fischeri. It's a cold adapted strain. And so the model, though I don't think this has been very well tested, is that at colder times of year and at greater depths, you'll see more Vibrio logii, and then at warmer conditions, you see more Vibrio fischeri. So it's interesting to think about how those 2 species may also coexist there.
That some dimmer and brighter strains might co-colonize, that's the same part. Could that be related to like how much light the squid is seeing from the moon, and then the squid could be kind of directing which strain that should be growing more or less?
I feel I should defer to Tim on this question. This is much closer to his expertise. I think that, you know, we don't have a great sense of, I presented at the beginning as this is how the counter-illumination works, but we actually don't know much about the mechanism there. So things that have been hypothesized are that the squid can move its ink sac around and provide a physical shutter to block light emission. And oxygen is a reactant in the luminescence reaction. So it's also been hypothesized that the squid can provision oxygen to the bacteria. I think these are really intriguing ideas. I think it'd be cool if they were tested. Yeah.
Okay, I'm gonna have something in molecular. But so on your CYP-F, do you know, do the BIN-K and RSCS both phosphorylate the same sites on CYP-F, or do they have multiple phosphorylation sites on the rec domain?
Yeah, we don't know. So I would say in Karen Weiss's lab has shown genetic evidence as to what the site is by which RSCS transfers a phosphoryl group to CYP-F. For BIN-K, I think there's a fairly new, we have bacterial tube hybrid evidence that BIN-K and CYP-F interact, but we do not have any evidence yet as to how that actually goes. Yeah. And we haven't had any success biochemically, like isolating the interaction. Like it's a total mess trying to in vitro phosphotransfer with these.
Yeah. We can talk. There's some tricks to like maybe make it a little better. Histidine kinases are really hard to deal with. I was gonna say though, do you know? So I guess you don't know if there's like more phosphorylation of the CYP-G than like more flexible. It's sort of okay.
Yeah. My favorite idea, which is probably wrong, is that the phosphoryl groups flow to BIN-K when it's inactive, when it's in the non-biofilm state. And then when BIN-K is inactive, like it's grabbing the groups off of CYP-F And then when BNKs shuts down, like in the squid, then instead the flow goes down to CYP-G. But the look you're giving me.
Also a kinase, right?
What's that?
Isn't BNK also histidine kinase from the?
Yeah, yeah, and phosphatase, presumably. Yeah, the look you're giving me telling me that's probably a
maybe.
terrible idea.
I think it's.
Okay, I'll try more. There.
How conserved are these genes across Vibrio? Like thinking about this case is beneficial, but in other cases there's like pathogenic. So do we see cases like this in other examples where their ability to biofilm might correlate with that?
Yeah, so I think the best example would be like Vibrio vulnificus, where that 18 gene biofilm locus, on average, there's about 70% amino acid identity, but it varies pretty much by which protein there is. So 17 of the genes are present and have orthologs. CYP-E, the one that was least conserved in Vibrio fischeri, it's a totally different gene in Vibrio vulnificus. It's another regulator, but it has no similarity at all. It's something we'd like to look into as to what the relevance of that is. Yeah, so it does use it. It's called the burp locus, and there it's but yeah.
So Mark, going back to the phospholipase.
Yeah.
So I think, if I remember correctly, Karen showed that the first histidine in CYP-F doesn't really contribute to signaling. It's just that histidine that's in the HPT domain.
Yes.
So, but in CYP-F of SR5,
yeah.
have you guys mutated that histidine, that first histidine, and see if that phospholipase is suddenly functional in SR5?
Yeah, so I think the first one is not important. So we think, and one complication is that I told you before in response to Rim's question that in ES114, right, you only need this domain. That's also true in CYP-F. Okay, so even though I didn't really show it here, the rect domain, the important domain for specificity is the D carrying domain here, but actually, if you just put in this domain, okay, that's you can still get it to colonize. And so our interpretation of that is, and actually you can put in the ES114 version of that too, and it's fine. So that's partly why our model is that in CYP-F, this domain is actively pushing phosphates down, whereas in ES114, it's actively pulling them up.
It looks like we're out of time. Give me one more time and thank you, Mark, for a wonderful.